A very good morning to all of you. Sorry, good evening to all of you. My name is Shivangi Suman, and today I welcome you all to this final day of workshop on mushroom spawn production. So, this is our third and final day. So, let's get started with our work schedule first. So, first, we are going to discuss that what was our work schedule. So this was our work schedule. We have achieved the first goal, that was the substrate preparation. After that, we mixed all those essential chemicals with our substrate on day two. Then on the same date, on day two, we did bagging, that is bag preparation, and after that sterilization process. So you saw that we yesterday we kept our substrate for sterilization, that is for the process of autoclaving. This is the third day, and today we are going to do the process of inoculation. After that, we'll keep it for the incubation. And at last, that is our goal 7, that is business aspects. So, in that, we are going to welcome one of our guests. He is going to tell you about everything that is needed in the business aspects of spawn mushroom production. Sorry, mushroom spawn production. So, basically, he will be dealing with that if you want to set up a plant, so what are the necessary things that you need, the price of the equipments, and the size of the room and all that. And along with that, he will be telling you about what are the certifications that you need for spawn production, as well as how to register the company and all other things. So I hope that this business aspect session is going to be really helpful for you, and all the participants who were requesting us and were telling us that we are not dealing with business aspects in detail, so we have kept that for today. I'm sure that session of 30-40 minutes is going to be really helpful for you, and after that we'll be taking the question and answer session so that you can ask about all your queries. Now let's discuss about what we have discussed in these 2 days, so that it can be a quick wrap up for things that we learned about. So first we dealt with mushroom production, introduction of mushroom. So what is actually mushroom? After that, we discussed about what is mushroom spawn. So how, what is spawn, how it is different from spores, and why we prefer spawn over spores for the commercial production. So that was the thing that we discussed. After that, we discussed about mushroom spawn substrate. So how for different mushrooms substrate differs, and why different substrates are used for different spawn. So we do not use one single type of substrate for every mushroom. There are different kind of substrates used for different spawns, but wheat is preferable, and it is preferable because, and we also discussed that why it is preferable, because for the growth of the mycelia, um, starch is the most important component, and wheat has 60 to 70% of starch. That makes wheat a really good component of substrate. It makes wheat a really good substrate, not component of substrate, but actually the substrate. Again, we discussed what are the reasons that make it a good substrate. It is nutrient rich, it provides good surface area for the mycelium to wrap around, it is easy to handle, widely available, etc., etc. After that, we dealt with that why we boil the wheat, or why we boil any kind of substrate. So we boil substrate so that all the nutrition that are needed by the mycelia to suck becomes available to them. So nutrient availability and hydration are all those reasons, and of course, removing impurity, etc., is the reason. For that purpose we boil wheat, or in general if we say we boil substrate for that reason. Now, after in second day session, we dealt with that once we have dried the boiled the mushroom, we have kept it overnight for drying, and after that, we are mixing all the essential chemicals that are need to be mixed. So we mix gypsum and calcium carbonate. So basically, gypsum maintains the grain's free flowing nature. Because of of course, we have boiled it. Though we have dried it, but there are chances that it can stick together. So to maintain that free flowing nature of our substrate, we add calcium sulfate, and to maintain the pH we add calcium carbonate. All these things were dealt in detail. After that, we discussed about the process of bagging in very much in detail. We discussed that why we need our bag to be filled only to two-thirds, how to make cotton plug, why cotton plug is necessary, and all that. After that, sterilization technique was dealt. Why sterilization is very important, as we have said multiple times during this workshop, that the major gap in spawn industry, that people are unable to produce good quality spawn, is because people have very low understanding of sterilization and contamination. So if you do not understand contamination, of course you will not be understanding sterilization better. So we discussed what is sterilization, then we discussed different kinds of sterilization, we discussed why certain kinds of sterilization is important for spawn industry and all that. After that, different methods of sterilization was discussed, and then we dealt about autoclaving. So you saw how autoclaving, the process of autoclaving is being performed. Along with that, you also saw what is autoclave, what is the working principle of autoclave, and then we saw the sterilization cycle. And even in the comment section yesterday, we had a lot of questions asking about that why all these three cycles, all these three steps of sterilization cycle is really important. So this was discussed, and this is a very important thing, this sterilization cycle thing. After that, we discussed about a case study. Now today we are this is the day 3 agenda, that what we are going to discuss. So we are going to take, we are going to start with the inoculation. We discussed the last lecture, we kept took a glance of that. Then we'll be dealing with the inoculation. After that, incubation. In the meantime, we'll be dealing with the theory along with it, and the most important section that is going to be covered about the mushroom enterprise advisory lecture. I hope this is going to be really helpful for you. And after that, we'll be taking the question and answer session. So before wasting any more time, let's get dive deep into the demonstration section first. So today we are going to start with cleaning the laminar. This is the laminar flow hood chamber. We'll deal about this in theoretical portion in detail later on. So in the demonstration section, we will see that how first we'll start with wiping of the laminar. So as you can see, we are opening the hood and we are going to wipe the laminar. So once you sit in the laminar, first thing first that you need to do is you need to wipe your hands with 70% ethanol. Why 70% ethanol? We'll discuss that also in detail later on. Then this is the Bunsen burner. You are going to, on this. And now we'll wipe the laminar cleanly with 70% ethanol. I hope the video is clearly visible to you. So while wiping the surface, we make sure that we wipe the surface really cleanly, because this wiping is a very essential technique. So make sure that you wipe the surface of your laminar properly. And we do not prefer any extra thing kept in the laminar. So keep only those things that are necessary for the experiment. We keep only those things inside the laminar, and anything else in that is kept outside to maintain the aseptic nature of the environment. Even once you are wiping the laminar, you make sure that once you have kept your hand inside, you're not going to take out your hand again and again. So we keep a beaker, a glass beaker or a plastic beaker, anything that you prefer, inside the laminar flow hood, so that all the tissue papers that you're using for the wiping, you're keeping it in that jar itself and not taking your hand outside again and again. And even after wiping, we do not keep anything on the surface. As you can see, it is wiped 2 to 3 times. Now, since we have wiped our laminar, we are going to take out the substrate that we have autoclaved yesterday. So we will take that substrate out from the autoclave and keep it in laminar and flow hood. So this is our laminar. The cooling has been done automatically. As we discussed yesterday also, cooling process is also needed. So you can see we are releasing the knob and now we are opening all other knobs. So even before taking out the chamber, we'll sanitize our hand, and so we do not open the chamber just outside the autoclave. We open the chamber near the laminar airflow so that just we take out anything, the thing that we have autoclaved, and directly put it inside the laminar airflow hood. So here goes our chamber. After sterilizing our hand properly, we open it, and we are keeping the substrate inside the laminar airflow hood. Now we are going to switch off the light, let the fan open, and we will on the UV. So why we are keeping it inside UV for? So because maybe there are some contaminants by the time we have taken it out from the autoclave and kept it in the laminar with that 2 to 3 seconds of time duration, there are chances that, and not chances, it's that there are certain kinds of microbes that has stuck on the surface of that ppe bag. So we need to sterilize that pp bag before opening it and then inoculating it. So these are the things that leads to contamination even more. So these minor things need to be kept in mind, and now we on the UV light, so it will do surface sterilization of it. So we'll wait for 20 minutes. So it's 5:14, and at 5:24 we'll switch off the UV light and we'll start the process of inoculation. In the meantime, we are going to take the theory class of what is autoclave and all that. So you can see our UV light is on, and our sample is kept inside the substrate bag, and we are doing this process for surface sterilization. So this demonstration you could see for 20 minutes. I hope it's visible on your screen. You could see it for 20 minutes. We'll keep it like this. And now let's move on with the theoretical portion. So in the theoretical portion, first thing that we'll discuss about what is laminar airflow hood. That will be our first slide of today's lecture. But before that, so the first slide is introduction of laminar airflow hood. So what is actually laminar airflow hood? It is a sterile workstation designed to provide contamination-free workspace for lab work. As you have, as you can see on your screen right now, that this is basically a workstation kind of thing. It's not a room, it's just a chamber that is used for contamination free workspace. So when you're working in laboratory and if you're dealing with any kind of biological sample, or even sometimes any kind of chemical samples, then these samples get contaminated very easily from outside sources, microorganisms. So keeping that safe from those microorganisms, if you want to work on any specific kind of microorganism, so you need it to keep it away from all different kinds of other microorganisms. And to provide that sterile aseptic environment, we use laminar airflow hood. Now it uses HEPA filtered air to remove microorganisms and dust particles. So the basic use of laminar airflow hood is that it uses a special kind of filter. That special kind of filter is called HEPA. Now that HEPA filter stands for high efficiency particulate air. So this special kind of filter removes all kind of dust particles and any microorganisms and spore kind of particles. All these particles are kept away so that the sterility of the environment can be maintained. Ensure sterile condition for handling sensitive biological materials. So as we said, any biological material if you're dealing with, because since any most of the biological materials are sensitive materials, so for dealing with them you need a contamination free environment, and for that and for maintaining that contamination free environment, laminar airflow hood is used. Protect samples and cultures from airborne contamination. I hope most of you, if you are from a life sciences background, you would have seen in your laboratories that whenever we are, making even media, so we open media after autoclaving inside the laminar flow hood, and the purpose of that is that it even get affected by airborne contaminations, because the air we are breathing right now, of course it has different kinds of microorganisms. Though we are immune to it does not mean the media will be immune to it. So any microorganism, if it will get a chance to come in contact with any kind of media, and the media will get contaminated. And of course other microorganisms, even if you're coming in, if you have a liquid culture of a media and you have inoculated that with a specific kind of microorganism, and if it is that inoculation process is done in the open room, because even suppose you have a small room and you think that this room we have performed other experiments, we have performed certain decontamination things, like we have performed fumigation in the room, and we are thinking that this room is sterile, but that's not possible. You cannot sterile a room because there will be airflow inside the room. You cannot control that, and that airflow will cause different kinds of microorganisms to move in and out of the cell. So if you are inoculating any culture in just in a room, of course that culture is going to be contaminated. However, you have sterilized the room, or you have performed any kind of sterilization in the room, it's really difficult to maintain that sterility. So for that shake, we need a chamber kind of thing. So it provides a controlled environment for reliable experimental results. So if you want reliable experimental results, you need chambers like laminar airflow hood, and is widely used in microbiology, biotechnology, pharmaceuticals, and mushroom spawn labs. So different kinds of labs. Either you are doing with, the main reason that mushroom spawn cultivation is not successful by so many people is because they do not maintain, those sterility things, and they do not focus on contamination better. So any kind of microbiology or biotechnology, any kind of biological data, if you're dealing with, you need to maintain the sterility. By the time you're dealing with it, even if when you are keeping it for incubation or you are doing the inoculation process, all that, for all those time, this sterility thing, the aseptic environment need to be maintained, and for maintaining that aseptic environment, we use laminar airflow hood. Now, this laminar airflow hood is very different from biosafety cabinets. If you think two of it is same, then let me clarify this, that the laminar flow hood is basically called a clean bench. Now, what does this clean bench thing mean? That if you are dealing with laminar flow hood, it means the purpose of the laminar flow hood is to protect the sample and not the user. So, if you are dealing with some kind of biological products like human cell line or animal cell line, or people who work with really pathogenic bacteria and viruses, or at the time of corona, you know, the scientists who were dealing with coronavirus for generating antibiotics, so they might not be doing, and of course cannot do it in laminar airflow hood, because the purpose of laminar flow hood is to protect the sample from contamination, not the user from contamination. So the basic difference of laminar flow hood and safety cabinet is the primary purpose of laminar flow hood is to protect the product or the sample from contamination. And if we talk about biosafety cabinets, it is used to protect the user first, and after that the protect product or the environment. So, and why, how these are designed to maintain that thing. So if you see, we are going to discuss later on that laminar flow hood has unidirectional flow of air. So that makes the sample, the product that you're keeping in it, it makes that environment aseptic for the sample. But if you're talking about biosafety cabinet, it uses a combination of directional airflow. So that keeps the user also protected and the sample also protected. And as we said, if you are dealing with something non-hazardous, where product sterility is the main concern, then you use laminar flow hood. But if you are dealing with something that pose a risk to the user and the environment, such as bacteria and viruses, or human and animal cell line, or viruses like coronavirus, then we prefer what biosafety cabinets. Now let's discuss what is the working principle of biosafety cabinets. Sorry, laminar airflow, because we are not dealing with biosafety cabinet right now. So we will discuss about laminar airflow hood. So a laminar flow hood is a sterile workbench that provides contamination-free environment. As we discussed, here it is also written. After that, if we deal with the working principle, it is air is drawn in and passed through a pre-filter that removes dust. So, if we want to understand the working principle, then we need to think it in three main parts. The first part is the blower and the or the fan. The second thing is the pre-filter, and the third thing is the HEPA filter. So, the what is the blower or the fan? So, if you, I hope you could see my cursor. So, above it there is a blower or a fan setup. So what does that blower or a fan does? This is a motor that pulls in the air from the surrounding room. So the purpose of blower or the fan is that it is basically a motor that pulls air in pressure from the surrounding room. Then there is a pre-filter setup in there. So this pre-filter is nothing but the first line of defense. So this first line of defense is a simple kind of filter, and it traps large particles, large particles of dust, maybe hair, maybe lint, and all those large particles. It is trapped by the pre-filter. And what does this do? This basically protects the HEPA filter. It improves the life cycle of HEPA filter, because if everything, even the heavy particles, are being trapped by the HEPA filter, it is eventually going to reduce the lifespan of the HEPA filter. So a pre-filter is set. This pre-filter is non-expensive. It's not that expensive. So it traps all those heavy dust, lint, and air particles, hair particles, if there are, because those are present in the environment. So it protects that. And next layer of protection comes the HEPA filter. So this is the main part that leads to this maintain the sterility of this laminar airflow hood. So we can say, as in human body, our heart maintains, keeps us running, in the same way in HEPA filter, in the same way in laminar flow hood, HEPA filter keeps the system running. So HEPA filter stands for, as we discussed earlier, high efficiency particulate air. And what is this filter? If we talk about that, what is this filter, then this filter is a dense mat of randomly arranged fibers. So, and this randomly arranged fibers is absolutely designed to trap 99.97% of airborne particles. Those are less than 0, greater than zero point 0.3 microns. So if any particle that is greater than zero point 0.3 microns or larger than that, it will be held by HEPA filter. And practically most of the microbes and spores and fungi and the dust particles, all these things comes under this zero point 0.3 micron size. So the chances of maintaining the sterility becomes really higher. And if you are dealing with anything of the biological sample or scientific sample, so we understand that we never claim that it is 100% safe. So that is why we say that it is 99.97% of chances of eliminating all kinds of airborne particles. Okay. So if you want to understand, the three main components, the first is blower, the second is pre-filter, and the third is HEPA filter. Now, we say that its name is laminar airflow hood. So what is basically laminar? Laminar is basically the way the air moves. So laminar flow, if you want to imagine, you need to understand about a free flowing calm river. So when you think of a free flowing calm river, what do you understand? The river, in every drop of the river, sorry, every drop of water that is flowing in the river, it flows in the same direction calmly. That what laminar means, that every, the air that is moving inside is moving in one direction, either vertically or horizontally, depending on what kind of laminar airflow hood you have. So it's moving, without creating any kind of turbulence. It is moving, as you can see here, these blue lines. It shows that it's moving in parallel lines, in a parallel straight line. So that movement of air, or that movement, is called laminar. Now there is also a different, and this laminar air movement helps in maintaining the sterility of the environment, because from the pressure, air comes from the top and it moves outside, because outside, because there at the bottom there are small holes created, and the air moves outside from there. Now there is a different kind of air that is called turbulent flow. So if the airflow is turbulent, it means some air is even backflowing. The pressure of air is very high, so it is getting mixed. Now this, what this laminar airflow does, that it's coming straight, it's moving out straight. So any microbe or contamination that is coming, it's in its line, or in its space, it's pushing it outside. But if the airflow is turbulent, so it is going to turbulent airflow, it means it's swirling around, moving inside this cabinet. So that will lead to contamination. Even some contamination can go outside, but even it can flow backflow. So that is why you need to understand that we so that is why when we are sitting inside the laminar airflow for work, so we keep our movements calm. If you're moving your hand very much vigorously, that will lead to turbulent airflow, and it can eventually lead to contamination. So that method is not preferred when you're doing performing experiments inside laminar airflow. Now if we take about, we discuss the process. Okay, what is the process? The first is, the whole process of this laminar airflow principle can be summarized in three very simple, steps. The first step is air intake. So air is taken in by the blower, then filtered by pre-filter, then HEPA filter. So first is intake of the air, second is filtration, and the third and the very important thing is unidirectional flow of air. So that is why it is called laminar airflow. So any kind of turbulence will create what will can lead to contamination further. So any vigorous movement of your hand or your body, or anything that is kept inside, is avoided when you are dealing with things inside laminar airflow. Then there is a very important thing that you need to understand, that is positive pressure, and how positive pressure prevents contamination. So what is positive pressure? If we discuss that, positive pressure basically means, positive pressure is a pressure within a system or space that is greater than the pressure outside environment. So basically, when we are working on laminar flow hood, so in chambers or in rooms where laminar flow hood is kept, in those rooms it is preferred to have ACs for cooling and not fan, because if you on the fan, it will create air pressure inside the room. So the air that is present in the room can backflow inside the chamber from the hood, because when you're working you have opened the hood, so from there the air can move inside and that can lead to contamination later on. So what is that positive pressure means? Because the air is moving in with a lot of pressure, and in laminar airflow movement, the chamber inside the, the pressure inside the chamber is way higher than the pressure in the room, and that maintains a positive pressure. And this positive pressure also prevents contamination, because if contamination, because if there will be backflow of air from the room, so and that air will enter from the hood side, not from the filter side, though of course it will not be filtered, because there is nothing inside the chamber for filtration. There is at the top, from where the actual air comes in, that part has the filtration thing. So if any kind of air or anything enters from the hood side, it will eventually lead to contamination. So this needs to be kept in mind, so that you prevent positive pressure, and for prevent maintain positive pressure. And to maintain positive pressure, you need to keep AC for cooling your room, in spite of using fan or any kind of table fan, that or cooler, that creates that disturbs that air pressure thing inside the room for laminar flow hood. Now our next slide we'll be discussing later. First, 20 minutes are over, so we are going to start the process of inoculation. Now we have surface steriled this substrate, and now we are going to move forward with the process of inoculation. So this is our media, in which we have spores of white oyster mushroom. This is our mother culture of white oyster. So I hope all of you understand what is mother culture. So basically we have inoculating the spores of white oyster mushroom. Spores of white oyster mushroom in media for using it as mother culture, or every laboratory has its own mother, I mean, they maintain their mother culture thing. You can use any other microbes, I mean fungus mother culture. Only the incubation temperature is going to be changed for that. Rest of the process will be similar if you are using some other mushroom else than white oyster. So first, when we sit inside the chamber, we will sterile the reagent bottle with 70% ethanol, after wiping our hand, of course. You can see that we are wiping the surface of the laminar flow hood again and again, because maintaining sterility is a very important factor. Now we are discarding the silver foil because it is not needed. We have on the now the mouth of the reagent bottle is sterilized with through with the help of flame, because that is very important. If you have done microbiology classes, you know that what is the method of inoculation. So that these processes are really necessary for maintaining the sterility of your mother culture. And any work that you do inside laminar flow hood, you make sure that you do it near your flame, because again it also reduces the chances of contamination, really. So the any process performed inside the laminar flow hood, it should be performed near the flame. That again helps in reducing the chances of contamination. And this is syringe. So we are going to take a sterile syringe, and then we will be taking our we are using sterile syringe to take media from our mother culture, the media in which the spores are inoculated. So again, you need to flame the mouth of your reagent bottle so that your mother culture does not get contaminated. And now we are pouring this spore inside our bag. Now again we'll tight it. As we discussed yes in yesterday's lecture, that your cotton plug needs to be neatly tied and it needs to be neat and clean. So now we are mixing the media that we have poured inside, and as we said in yesterday's lecture, that you need to lightly tap after inoculating. You need to lightly tap your substrate so that your mycelium gets completely mixed. Now again, before closing this, we need to wipe the surface for maintaining the sterility for next use. Wipe your hand. And performing any process, any experiment inside laminar airflow hood, you need to wear gloves. So now you can see our substrate is being inoculated by the liquid media that has spores present in it, and we are lightly tapping it to mix it well. Now the next thing is that, I hope you could see it in zoom. After inoculation, the next thing that we do is we do the process of incubation. So we keep the substrate inoculated by the spores, by the mycelia into the incubator and maintain the temperature. So for spawn production, we need not to maintain the light or the humidity. We only need to make sure that temperature is maintained properly. So we'll see about the things about the incubator now. So there goes our incubator, and we have set the temperature to 26.6 degrees Celsius since it is white oyster mushroom. So for different mushrooms, the temperature is different. And now we kept this bag inside the incubator And we have set the temperature. As we said, humidity or light is not the factor for spawn production. So we only have to maintain temperature. Now we have maintained the temperature. So this was the demonstration part. Now again, before closing the laminar airflow hood, we need to decontaminate it properly, sterile it properly with the help of 70% ethanol. And the syringe filter that we have used once for inoculation purpose, we cannot reuse it. So we are going to discard it. And now we will on the UV for 20 minutes to sterile the surface before the next use. So this was the demonstration part. I hope you understood that how spawn production takes place actually. I hope you would have seen everything about spawn production better. So now please also understand the theoretical parts, because people go and attend seminars and they see how it is being done, but since they do not understand all the basic and the essential parts of it, means you do not understand how contamination is being caused, or how you need to sterilize certain thing, how you need to see that your substrate has right quantity of water or it is over wet. So all these things need to be understood, and for those things either you need to study or you need to attend lectures properly. So let me deal with it as soon as possible, so that we can get into the business aspects early on. But before that, let's discuss with all these things uh, fast. So before we discussed about the working principle of the laminar flow hood, I hope you understand what is positive pressure also. Now there are two types of laminar flow hood: vertical and horizontal. So the second image is of vertical laminar airflow hood, and the laminar flow hood you saw right now was also vertical laminar airflow hood. So in vertical laminar flow hood, basically the fan or the HEPA filter or the pre-filter, all these things are located at the top. So the air is drawn in and filtered and then flows downward, and this downward flow helps work on this work surface पे that downward flow of air moves. So what are the advantages of, if we talk about vertical laminar airflow? So it provides space efficiency. If you have small space and you want to set up laminar airflow hood, so since it is drawing air from the top, you can also set up in small places. Again, it provides safety to the operator. So since if you are dealing with something that has powders or fumes or some volatile compounds, and if you are dealing it in horizontal airflow laminar hood, so the air comes from where? From back end to the top, so it will come to you, and all those volatile particles or the powdery thing can blow towards you. So it actually threatens the safety of the user at times. So this is also the reason, these are the advantages of vertical airflow. This causes less cross contamination, and if you want to accommodate larger equipment, so you want to keep inside the vertical laminar airflow hood, then vertical laminar airflow hood is preferred. The only thing that is the disadvantage of a vertical laminar flow hood is it causes airflow turbulence at times, because it is taking greater space, from top till the bottom to move, and anything that is kept inside, if you have kept something like reagent bottle and all that, so the air strikes those surfaces, and later on, I mean, while it is striking those surfaces, it can cause to air turbulence. So this is one problem with vertical laminar airflow hood. If we discuss about horizontal laminar airflow hood, so the HEPA filter is positioned at the back of the cabinet. I hope you could see there HEPA filter is positioned. So air is drawn in, filtered, and then flow horizontally across the work surface towards the operator. The advantages of this is that it creates less turbulence and it's superior for sample protection. If we compare vertical and horizontal, then horizontal is superior for sample protection. And if we are talking about the disadvantage, the major disadvantage of it is that it expose the operator to those hazardous substance, because the air is moving from backward to front, and anything, maybe you're working with nanoparticles, then those nanoparticles can move towards you, and maybe you inhale it and it can cause certain problematic things. So that is a problem. And the second problem with this is it requires more space to be kept, the horizontal laminar airflow hood. So these are the two disadvantage. But if you say that which is preferred or which is better, then none of it is better or something. If you have small space and you want to keep user safety in mind, so you should prefer vertical. And if you have larger space and you need to work better with less contamination, so you can prefer horizontal laminar airflow hood. Two of which is good, so you can use any. Now this is about types of laminar airflow hood. Now if we talk about importance of UV light, as you have seen, we have on UV light for 20 minutes after keeping the substrate and substrate bag inside. So basically, why this UV light is used in a laminar airflow hood. So I hope you understood. This is done because we want to decontaminate the chamber. But how this decontamination takes place, if we talk about that, so the UV light that is emitted inside this is UVC light, and it has a wavelength of around 254 nanometer. So this UVC light at 254 nanometer actually damages the DNA or and the RNA of the microbes that is present on the surface. One thing that you need to understand very clearly, that suppose you have media inside a reagent bottle, and you kept that reagent bottle along with the media inside it in the laminar airflow hood, and now you have on the UV. So the UV light is only going to affect the microbes that are present on the surface of the reagent bottle. It is not going to penetrate inside the reagent bottle and sterilize the media that is present inside it. We have sterilized media using autoclave, and now we are keeping it in UV for 20 minutes, just to for the time duration we took to take out our reagent bottle from laminar flow I mean autoclave and keeping it in laminar flow hood. That 3-second gap would have caused a lot of microbes to get attached to its surface. So we are just killing those microbes. We are not sterilizing the media inside it. Similarly, it happens for everything that we are keeping inside. So if we are keeping the substrate bag inside, the substrate is already decontaminated using the autoclave method. Now it is kept inside the UV for decontamination of bag, because maybe there are certain microbes that would have stuck on its surface. So we are keeping it for sterilizing that. So basically, it damages the DNA or RNA of the microbes and prevents it from replicating. Of course, if it is damaged, it's not going to replicate and eventually it dies. So the main purpose of UV is decontaminate the work surface and hood's interior before and after work session. You would have seen that once we wiped the surface and we kept the substrate bag inside, we on the UV. So those it was not just decontaminating the UV, the substrate bag, but also the surface of the chamber. That is also very important, that you need to decontaminate the surface of the chamber, because if you're not uh, you know suppose we have as you have seen that at last we have wiped the surface clear very well with 70% ethanol, and then we on the UV. Why we did that? Because there are chances that maybe there are some microbes that would have, when we were taking out the spores via using those using syringe. So there are chances that spores are very fine particles. Then some of the spores would have flown and stuck on the surface of the laminar flow hood. So when you on the UV light, the UV light kill all those microorganisms that would have stuck on the surface while you were doing that process. So when you come again and now you're dealing with some other microorganisms, so that cross contamination can be prevented. So this needs to be understood. Now one thing that is very important and you should know about is UV light is very dangerous for human beings. So it is really hazardous for our skin and our eyes, and it can cause real damage to it. So whenever you are using UV light, you will not on UV light while you're working in your laminar flow hood. So once you have completed your work, then you on the UV light for the sterilization process, and after that you will again switch it off. So never get in touch with the UV light. It can be cancerous. It has been proven that it cause cancerous. It can cause cancer later on. So do not keep yourself exposed to UV. And this procedure takes, generally we on UV light for 15 to 30 minutes. So we have kept it on for 20 minutes right now, as you have seen, but the time duration for sterilization is around 15 to 30 minutes. It can vary. It can keep, you can keep up to 30 minutes, but 20 minutes is also fine. Now the applications and safety precautions. So as you have seen, what is the application of UV? Sorry, laminar airflow hood. So you are dealing with any kind of any kind of biological sample or any kind of chemical sample which has chances of getting contaminated. We use laminar for that purpose. So tissue culture and microbiology labs, in mushroom spawn preparation and production, you are seeing it right now. Pharmaceutical and biotech industries handling sterile medical devices, all these things are done inside laminar airflow hood. Now precautions that you need to take: turn UV light off while working, harmful to eyes and skin, as we discussed right now. It is very important to understand. Always disinfect surface with 70% alcohol before use. This is the thing that we need to discuss quickly. That as you have seen that even when we boiled the substrate, and we were mixing the substrate with the chemicals, then we wiped the top which was used for those mixing process with 70% ethanol. And every time you see we are doing anything, we are wiping our hand with 70% ethanol, we are wiping the laminar chamber with 70% ethanol. So why 70% ethanol? So basically this 70% ethanol, it denatures the proteins and dissolves the lipid membrane of the microorganism, that may, even if the microorganism is present inside, or bacteria or fungus, anything that is present. So it basically denatures the cell wall and eventually kills the microorganism. Now, if a question comes to your mind that why 70% ethanol and why not 100%, because 100% will be even more effective, but 100% is not that effective, because we know that alcohol is very alcohol is very volatile. So if you're using alcohol, then 100% alcohol will get evaporated very soon, and it will not get the time, because it evaporated early. It will not get the time to denature the cell wall and eventually kill the microorganism. But in 70% ethanol, 30% of water is mixed. So that 30% of water slows down the rate of evaporation. So this gets enough of time to kill all the microorganisms and make the surface sterile. So that is why 70% of alcohol is used for sterilization process. This is a very good disinfectant and used in laboratory, all kind of laboratories. So you need to understand this. Work slowly inside the chamber to avoid airflow disturbance. As we have discussed, that laminar airflow is straight parallel flow of air, and any kind of air turbulence can cause backflow of air, and it will eventually lead to contamination. So when you are working inside the laminar airflow hood, do not move your hand very vigorously, or the things that is kept inside very vigorously. Do it easily, keep yourself easy, and do things calmly. Do not block airflow grills with instruments. So do not, if you have horizontal laminar airflow hood, so we know that the air moves from backward to front. And if you have kept a lot of instruments at the back of the at the back side of the wall or the laminar flow hood, so eventually it is going to obstruct the movement of the air. And even it is advised to not stand very near to the head or hood of the laminar airflow hood. So the hood that you're using for opening and closing the chamber, it is advised to not stand very near to it. One thing more is advised that when you're sitting inside the laminar airflow hood, you're not advised to speak. Why you're not advised to speak? Because if you're speaking, there are certain microbes inside your mouth, so it can go and contaminate your sample. And always when you're using laminar airflow hood, make sure that you're wearing mask and gloves. So these are the things that you need to keep in mind. Now moving towards the next slide, we have introduction to incubators. So what is an incubator? You have seen that we have kept our substrate after inoculation to the incubator. So an incubator is a device that provides a controlled environment for the growth of microorganisms, plants or animal cells. So basically, it provides a controlled environment where you can either maintain temperature, pressure, and all that. Sometimes pH also. So maintains constant temperature, humidity, and sometimes CO2 and O2 levels. On advanced incubators, you will see that it along with temperature also maintains humidity, carbon dioxide, and oxygen levels. And this is important for microbiology, biotechnology labs. Why it is important? Because in the wild some microbes grow on 20 degrees celsius, others on 30 degrees celsius, but our environment fluctuates. We have right now temperature around 32 degrees celsius, so any microorganism that grows in 20 degrees celsius cannot grow in 32 degrees celsius and our normal room temperature. So for that we need incubators to maintain the environment. Now, if we talk about working principle, works on the principle of temperature regulation to mimic natural growth conditions. As we said, heating coils and thermostat maintains the desired temperature for maintaining the desired temperature. Some advanced incubators include humidity and gas control system. Humidity is very important to be maintained, ensures optimal condition for reliable and reproducible growth. Applications again, microbial culture growth, tissue cell culture, mushroom spawn development, biological testing and research. The things that you need to keep in mind: do not open the door frequently, because if you will open the door frequently, it will disturb the temperature. Regularly disinfect and keep incubator clean. This is a very important thing. Avoid overloading, ensure proper air circulation. So please do not keep a lot of things together inside the incubator. Monitor temperature with an inbuilt extra thermometer, and even your display is showing the temperature. Sometimes for checkups you can use extra thermometers to check the temperature. That will help in keeping in mind the calibration track of your instrument also. Now if we talk about incubation and growth duration, so we have mentioned here two mushrooms, that is button mushroom and oyster mushroom. So today the mushroom we have inoculated was oyster. It was white oyster mushroom, and we have set the temperature 26.6 degrees celsius, as you have seen. So for oyster mushroom, the incubation temperature is 25 to 28 degrees celsius, and spawn times, you can see that spawn will get developed within 10 to 20 days, sometimes as fast as 7 to 10 days. And for button mushroom, incubation temperature is 23 to 26 degrees Celsius, and spawn get developed within 14 to 18 days. Sometimes up to it takes 25 days. Now you can see that these days are very much variable. So why it is variable? Because in the wild the scene is very different, but when we are doing it commercially in our laboratories in lab setup, so the best amount of temperature that you can provide, the better will be the growth. If your temperature fluctuates and varies very much, it is eventually going to hamper the growth of the spawn, because mycelia need a specific temperature to grow and replicate itself. So if your temperature is fluctuating very much, it is going to hamper the growth of mycelia eventually. So you need to keep that in mind. So why, what is the reason behind maintaining the optimal temperature? Because of course mycelia would get affected, and if you're not maintaining, why you need to maintain the temperature? So if you're not maintaining the temperature better, your growth of mycelia is going to be delayed, and we know if the growth is delayed, the chances of contamination is very higher. Now we need to understand that what is the relationship between the temperature and time. So the better you provide the time, the lesser temperature, the lesser the time it will take. But beyond temperature, there are certain more things that need to be kept in mind. That is substrate moisture content. If the moisture content of the substrate, that on which you have growing the mycelia, if it is very high or if it is very low, it is going to affect the growth of mycelium, and it is going to hamper the growth of mycelium, both more wet or more dry. So that is why we said time and again that check the moisture of your substrate. And the next thing that you need to understand, that the amount of and the quality of the mother culture that you are using. So two of it is very important: the moisture of the substrate and the quality of the mother culture that you are using. If you are using poor quality of mother culture, eventually it is going to be lead to poor quality of the spawn. So make sure that you are using good quality of mother culture, buying your mother culture from a really reliable and nice place, so that it can lead to good quality of um, good quality of spawn growth later on. Now we are, as you have seen, that we have completed our two of the goals, that is incubation. I mean, sorry, inoculation and incubation. Next, we are going to start with the business aspects of mushroom small spawn plant setup. And for that, I want to welcome Mr. Shubham Maurya, Mushroom Enterprise Advisor and Director, Academic Monk Private Limited. So, sir, please, I welcome you. And now, sir is going to tell you about all the commercial aspects of the mushroom cultivation. I hope you will enjoy. So, sir, welcome. Please continue.
Thank you so much, Shivangi, for the introduction. Hello, everyone. Welcome to our session. Today, I will walk you through the essential steps of setting up a mushroom spawn production plant, which will include cost, space requirements, production estimates, and registration process. When setting up a mushroom spawn facility, there are three key factors to consider: cost, space, and production capacity. Understanding these will help us plan effectively and run a sustainable operation. The total setup cost ranges between 1.5 to 5 lakh rupees, provided we have already have a building setup. The major requirements includes an autoclave costing between 40 to 80,000 rupees and a laminar flow which will cost you around 60,000 to 1.2 lakh rupees. We are giving you the range of minimum and the maximum as well, because there are different companies which are selling the machines, and you, depending upon your availability and your area setups, it will cost you different. Okay, so we, and the major essential which we would require are the refrigerator, air conditioning unit, and the monthly consumables, which will be each refrigerator, air conditioner, and the monthly consumables will range you around 20,000 rupees to 50,000. Additionally, we will require 1 to 2 semi-skilled workers. Whose salary will be around 10,000 to 20,000 per month, depending upon the daily wages or the wages law in your state as well. Of course, these number will be variable for each and every state in India as well, or anywhere if someone is going outside the India as well, or in the abroad countries, the daily wages they have to pay on their own country setup as well. So in total, you can say it's a cost, matlab, it's a very cost effective process. This whole cost doesn't cost you so much, because ranging these things, it will, the total investment of a person who is doing this business will cost you around 8 lakh rupees, something like that. Okay, depending upon the region where the person is residing. The most important thing in this area in the production capacity in terms of space, this whole setup requires around 250 to 300 square feet of area. This area will include the media preparation, the sterilization room, an inoculation room, an incubation space, and a small office and storage area. With this setup, you can have at least a production capacity of 150 to 200 bottles per day, or a bags in which form you are settling it in, which will total around 4,000 to 6,000 per month. The total revenue which you can generate from this whole setup is around 1.2 to 2.5 lakh rupees per month, in which the break-in period is about 8 to 12 months. Okay, so it's a good business in terms of numbers. In small setup, you will have a small setup and you can earn a lot of money through that, because when you will complete your break-even period, then you can earn a lot of money in that, and that will all will be your profit. So let me tell you about how you can you register this business in this whole in, and currently talking about the Indian setup. So you have to register as a private limited company. There are many type of companies. There are LLP, there are proprietorship, there is a one person company as well. But I will suggest you to register your company as a private limited company. Because if you want to settle this business, for example, if the whole setup cost is around 7 lakhs to 8 lakh rupees, you can get this amount through government of India as well. But the government of India right currently they are providing this amount for the startups only. Okay, so for startup you need to register as a private limited company. So for the startup, you have to decide the name of your entity. You will have to obtain the digital signature, and you will get the DIN number as well. DIN stands for Director Identification Number, and DSC stands for Digital Signatures. Okay, so you have to contact a CA, a chartered accountant who will register your company on the MCA portal, and he will be providing you all the things. The cost of this company registration comes around rupees 15,000, in which you get each and everything. Your company registration will be there. You will be provided with the pen. You will be provided with the GST, MSME. All the things will be provided to you in terms of the company, which will help you to in getting the startup registration. And once you get the Startup India registration, you can also register in the Start in UP registration for talking about the people of UP as well. Okay, from Uttar Pradesh or from the different states, they can register in their own state startup contributions as well. So because government of India and states are providing different policies for the students, those who are the famous entrepreneurs who wants to do the business, they help in settling the business. Currently in Uttar Pradesh, let me take the example of Uttar Pradesh as we are residing here, our companies here. You can get around 5 lakh rupees of prototype fund or setting up a lab or anything, any business model which you are coming with. So you need to have a presentation and you need to provide that presentation, and then you have to get incubated as well. ठीक है, incubation provide you the मतलब basic strategies they divides for you. And for that you need, like, as you are making up as mushroom spawn production, it involves agriculture and microbiology as well. So some approvals will also be required. So this includes the GST registration. You will be needing the pollution control permissions for larger operations. Not for the small operation, you will not require this pollution certificate and all. But for small scale production, you will not require. But for the large scale, you will be requiring this pollution control. And in some cases you will be requiring the drugs or pesticide licenses. This will depend on state to state, which states are allowing you to do that business. For example, in UP you don't require this drug or pesticide license for spawn production, but you will be requiring the pollution one. Okay, so basic requirements of documents which you will require are Aadhaar card, PAN card, your proof of business address, passport size photos, depending on the entity type. But for the private limited company, you will be requiring the, if you don't have your own business, then you will require this landlord agreement, rent agreement with you with the amount of 5% in that. So these are the basic requirements which you have to fulfill for getting the company registered, and you can start your business. These are the basic things which you will require. So all the things which whatsoever you will buy or purchase, any machines or any machinery which you are keeping in your business, just try to get the good quality of machines and maintain the cleanliness of the laboratory, which is very important for the spawn production. Because as you know, good seeds leads to good harvest, but bad seeds results in failure. Okay, and always purchase your mother culture from the trusted companies only. Do not buy from anywhere that they are telling us that we sell it, but try to verify from where you are getting. As you know, Flavinlabs is also sell the do the spawn production, and they also sell. You can buy from them as well, or any other entity which you trust upon. So these are the various things which you can go, and your popular spawns like button mushroom, oyster mushroom, cordyceps, and there are many other varieties as well. And if you have any queries related to the setup of this whole spawn production unit or a mushroom unit as well, so you can contact Flavin Labs Private Limited anytime. They are going to help you, and they will provide the full project report for you, and they will also tell you the ways to get the funds from the various different organization or from the MSME as well. So this all is the whole process for this. And I would like to, Malab Shivangi would continue about the things as well.
So I hope all of you would have understood about that how you can set up your mushroom spawn production plant. This was the query with so many people. They have asked again and again that we are not dealing with it. So I hope most of your queries are being dealt here. And if yet you have some problem, you can DM us. You can DM our admin about this, and he will be dealing with your queries free of cost. So you can do that with us. Along with this, I am sure that if you have any problem right now also, our advisor is with us right now, so you can post your questions in the comment section. Now we are going to take questions, so please keep posting your questions, and let's deal with your questions one by one.
First question is, where is the mother culture kept preserved?
The mother culture is kept preserved in the deep freeze. So, freezer is used for preserving mother culture.
Another question is, please explain how can we activate the mother culture?
So, if you already have mother culture with yourself and it has dried out, and you are unable to activate it in liquid media, then you can use sugar solution for it. Glucose solution is used for, it is kept in 20 minutes in glucose solution, and after that kept in the liquid media and it get activated. And if you are asking that you do not have mother culture and you want to inoculate fresh mother culture, then suppose you want to inoculate, you want to have a mother culture of button mushroom, then you can buy butter mushroom from the market, and cutting it from the inside, taking a sterile portion, you can inoculate it in the media, but it's a tough process. Tough, not, it's a tedious process. So you will have to do it again and again for getting the pure culture.
Next question is, please also explain how the mother culture is made.
As I said, you can buy any kind of button. Suppose you are buying button mushroom from the market, you're washing it properly, sterilizing it means you're cutting it. And taking flesh matlab tissue from inside, and then you can keep it in the media and then incubate it. You will get the mother culture, or you can buy mother culture from some trusted source.
Next question is, do you also culture blue, yellow, and pink oyster mushrooms?
Yes, we do culture and we also sell, so we provide blue, yellow and pink oyster mushroom. You can connect with us for that.
Next question, which culture media is used for culturing?
Liquid broth is used for, and the broth you have seen right now was PDA, I mean PDB, potato dextrose broth.
Next question, please explain how the mother culture of button mushroom is prepared.
So buy a button mushroom from the market, sterile it properly, wash it, then wipe it with 70% ethanol, then set in the laminar airflow hood. Use a sterile scalpel, buy new sterile scalpel. Cut the mushroom, from the bottom. Then take out, prepare the media PDB, because liquid media is used for, doing the culturing in the start. Now sit in the laminar flow hood. You can take out a sterile piece of tissue from inside of the mushroom and you can inoculate the media. After that, your media, in 6 to 7 days you can see your culture has prepared. Then you have to do reculture and it again and again, because there are higher chances of contamination on first time of inoculation. So this is the whole process.
Another question is in Hindi.
Hmm.
मैम laminar airflow क्यों important है?
क्योंकि लैमिनार एयर फ्लो स्टेराइल इन्वायरमेंट इंट्रोड्यूस करता है। जैसे कि अगर आप इस रूम में बैठे हैं और आपको मशरूम स्पॉन का कल्चर बनाना है, तो अगर आप इस रूम में बैठ के मशरूम को काटेंगे और वहाँ से एक टिश्यू लेके उसको उसमें डालेंगे, मीडिया में डालेंगे, तो पहले जैसे ही मीडिया एयर के कांटेक्ट में आएगा, एयर में बहुत सारे माइक्रोब्स हैं, वो सारे माइक्रोब्स उसमें चले जाएंगे और धीरे धीरे ग्रो करने लगेंगे। तो इवन दो आपने मशरूम को सही से भी डाला है उस मीडिया के अंदर, कंटामिनेशन ऑलरेडी अंदर जा चुका है, बिकॉज हमारे एयर में बहुत सारे कंटामिनेंट है। अब लैमिनार फ्लो हुड के अंदर लैमिनार वे में स्ट्रेट पैरेलल वेज में एयर पास हो रहे होते हैं, ऊपर से सीधा नीचे की तरफ बाहर निकल जाते हैं या सामने से बाहर की तरफ निकल जाते हैं। तो जितना भी कंटामिनेशन होता है वो बाहर आ जाता है, कोई कंटामिनेशन सरफेस के अंदर रुकता नहीं है। तो अंदर का एनवायरनमेंट बिल्कुल स्टेराइल एसेप्टिक एनवायरनमेंट होता है, जिसकी वजह से आप कोई भी बायोलॉजिकल डेटा या फिर केमिकल डेटा जो बहुत जल्दी कंटामिनेट हो जाता है उसके साथ डील कर रहे हैं, तो वो डीलिंग में कंटामिनेशन के चांसेस जो है वो 99.97% से कम हो जाते हैं। तो यही रीज़न है कि लैमिनार एयर फ्लो बहुत इंपॉर्टेंट होता है अगर आप लैबोरेटरी वर्क में इन्वॉल्व हो रहे हैं तो।
Please explain the difference between UV light and sterilization.
So, sterilization is the process of making the environment contamination free. Sterilization, UV rays is not different from sterilization. UV ray is a type of sterilization. So, if you want to ask the difference between autoclave and UV rays, then autoclave is doing what? It is sterilizing. If you are preparing the media, now if you're keeping it for autoclaving, the process for the process of autoclaving, then in autoclave your media is being sterilized, and along with the media your bottle is also being sterilized. But now you take out that bottle, reagent bottle, from the autoclave, and you're keeping it inside your laminar airflow hood. Now you have on the UV, so any microbe that has stuck on its surface, by the time you have taken it out from the autoclave and kept it inside the laminar, उसके बीच में अगर कोई भी माइक्रोब उसके सरफेस पे जाके चिपक गया है, तो जब आप यूवी लाइट ऑन करेंगे, यूवी लाइट उस सरफेस पे जो माइक्रोब्स प्रेजेंट है उनके स्किन के थ्रू पेनिट्रेट करके उनको किल करेगा। तो यूवी जो लाइट है वो सिर्फ सरफेस स्टरलाइजेशन करता है। जो ऑटोक्लेव है वो इनसाइड, मान लीजिए कि व्हीट ग्रेन के इनसाइड जाएगा और उसके अंदर भी जो माइक्रोब्स है उसको भी किल करेगा। सो यहाँ पे यूवी लाइट से मीडिया स्टरलाइज नहीं होगी, सिर्फ बॉटल स्टरलाइज होगा। तो इफ़ यू वांट टू डू सरफेस स्टरलाइजेशन, then UV rays is preferred. But if you want to do complete thorough sterilization, then we do autoclaving.
Next question is, please explain the difference between oyster mushroom mother culture and button mushroom mother culture.
These are two different kinds of mushroom. So of course their mother culture will also be different. So for oyster mushroom mother culture, you will retrieve that mother culture from oyster mushroom. For button mushroom, you will retrieve that mother culture from button mushroom.
Next question is, why is 70% ethanol important?
Because this is used for surface sterilization. So once suppose you are doing, you have to wipe, I mean, you are mixing your substrate with the chemicals. So before that, what will you do? You will decontaminate, you will clean your surface, and it cause better penetration. So 70% ethanol better penetrate through the walls of the microbes, kills it, and make the surface contamination free.
Next question is, what is ideal temperature and humidity for incubation and mushroom cultivation?
So different mushrooms incubate at different temperatures. So you can see the chart is available on Google. And also we have shown in our presentation that for button mushroom, it's for example, for oyster it is from 25 to 28, for button it is different. As you have seen, we have set the temperature to 26 degrees Celsius. So for different mushroom, different temperature is used for incubation.
Next question is, how long does the incubation period usually last for common mushroom like button, oyster, or shiitake?
It is different for different mushrooms. For example, if you are talking about oyster mushroom, it takes 10 to 20 days. Sometimes, if you are providing consistently 27 degrees Celsius or 26 degrees Celsius, then it takes 7 to 9 days for the spawn production. And similarly, if you are talking about button mushroom, it takes about 14 to 18 days. So, if you are providing consistent temperature, it will take 14 to 18, and oyster will take about 10 to 20 days.
Next question is, is it not possible to make your own mother culture?
Yeah, it is possible if you know. I mean, if you are from life sciences background and you know how to do, the culturing things, you have done it in your laboratory, then you can do it. But if you have not done it again and again, then it's really difficult, because retrieving the sterile tissue from a mother culture or any mushroom sample becomes really difficult. And even for making the pure mother culture, you need to have a fruiting body. So buy the fruiting body from the market, and then if you can culture it, then you can culture it.
Next question is, someone is asking, is it necessary to use autoclavable polythene bags or normal? We can use normal poly bags.
No, actually autoclave has a temperature of 121 degrees Celsius. So if you are going to use normal polybags, it is going to get melted, so you cannot use. You will have to buy autoclavable bags for that, because the temperature and pressure is really high inside that.
Another
So
question is, why incubation rooms be kept dark and undisturbed during this time?
because spawn does not need light for growth, and if you are moving in and out time and again, then it is going to make the environment disturbed. And of course it also leads to contamination later on, so that is why it is kept dark, to maintain all those kinds of disturbances. And since spawn develops better in the dark, so this is also the reason that it is kept dark.
Next question is, what changes indicate that the incubation stages is complete and growth can begin?
We have 1 minute more, so please keep posting your questions so that we could answer more and more. And you can also ask in the chat.
Next question is, what changes indicate that the incubation stages is complete and growth can be begin?
In fruiting body.
Yeah.
Yeah. So if you're talking about fruiting body, then if you're talking about white oyster mushroom or about pink oyster mushroom, so if the fruiting body of pink oyster mushroom has developed properly, then you can see it has turned pink. And how you will know that it is not getting over incubated? Then if it is getting over incubated, that you can see the formation of spores, as well as it will start turning gray. So once it has become full blown, it will come to its original color. So that is why we come to know that it has now come to its.
Next question is, how does the duration of growth vary among different mushroom cultivation and with respect to fruiting growth?
Different mushroom has different time duration for its growth, and of course different temperature also. So if you are talking about if you're talking about oyster mushroom, it's 70 days. For button also, it is 70 days. So 70 days is the time duration short of for the growth of fruiting body in any kind of mushroom.
Next question is, which factor affect the duration of mushroom growth and yield?
Temperature, and along with temperature, moisture, and then the light. So these three factors affect the growth of mushroom.
Sample we can buy from your lab.
So someone has asked that what is the minimum sample size that they can buy from our lab. So you can start buying from 10 kgs. Again, there is, I think there is. So is there any more questions that you want to ask?
इन हिंदी, मैम मीडिया को कितने अमाउंट में पोर करना है?
मीडिया को कितने अमाउंट में पोर करना है? अगर आपका पूछने का मतलब ये है कि अगर हम कितना इनोक्यूलम साइज डाल रहे हैं, तो अगर आप 100 ml मीडिया ले रहे हैं तो उसमें 5 to 7 ml हम उसमें इनोक्यूलम साइज जो हमारा होता है वो 5 to 7% का होता है. So so I think there is no more questions left. So this is the last of the session. I hope you would have learned whatever you have here come to learn, and I hope कि आप community का part है. So be in the community. Do not leave the community, because we'll keep posting about different updates of life sciences. We keep posting that in our community, and I hope you would have learned the things that you are here to learn. So I am sure now you understand what is spawn, how spawn production is done. And any one of you, if you want to set up a spawn production plant, you can consult with us. And if you're consulting on phone, it's free of cost, so you can ask your queries there also. If you have any more questions left, you can keep posting in the community or in our group, and we'll answer you there. Our certificate is available, so if you think that the session was worth and you should take the certificate, you can apply from here. So please apply from here and take the certificate and make this learning valid. So whenever you go somewhere, or there are certain you know uh, places where you go and you say that you have learned something, so you need to provide the certificate. So if you think that certificate is worth, please take the certificate and be a part of the community, so that any more updates about the life sciences that come, you are updated about it, and you can join other sessions as well. So we keep most of the times our session free of cost, so anyone who wants to join it for the learning purpose also, they can join. So the tagline of Flavinlabs, that we have learned and got a lot from the science and the science faculty, so we want to return it to our people and to the science and the students, so that they can learn and grow better in their life. So happy learning, keep learning. I hope you would have enjoyed the session with me. If you have any comment that what can be improved or something that you have not understood, please write in the feedback form. We will try to cure that in our next sessions. So thank you so much, everyone. You have been a great audience, the questions were nice, and let's meet sometime in some other session. So thank you.